So I'd like to thank the organizers for inviting me to talk today. I think I am the last talk, so I'm the only thing keeping you from coffee, probably. So I was asked to speak to you about some of our work on with Burkholderia phages and our attempts to engineer some of these phages. So I'll try to keep in time, and so we can all end on time here. So this is a phage meeting, so I don't need to go too much over the life cycle of the bacteriophages. So in this talk, the temperate phage, the lysogenic pathway is going to be relevant, right? So we're going to be talking about a phage which actually is a temperate phage, so it's able to enter both the lytic and the lysogenic pathway. It's a pretty standard temperate phage. It's able to incorporate into the host chromosome through an integrase, and we do know the attachment site for that. But it has some very interesting features in its induction. So we were interested in using phages as therapeutics. We've been involved in attempting to get phage collections for Burkholderia infections in CF patients for some years now. And in general, temperate phages are not really preferred for therapeutics, and there's really two reasons for that. Number one being that temperate phages will form a lysogen. And the lysogens, of course, are immune to further infection by the phage, and you get lysogeny often at relatively high rates, even in the low percentages of infections. And so you very rapidly accumulate a population of lysogens of the phage, which are then immune to the phage, and then the phage will stop working. And another reason why temperate phages are generally not preferred for therapeutics is that they can sometimes carry virulence factors in their genome. So many temperate phages carry what's called morons. And these are accessory genes. They're often actually expressed from the prophage state on their own little promoters. So they have their own little transcripts. And these can be toxins or things involved in immune evasion or antimicrobial resistance. Some famous versions of this are, for example, Shiga toxin or a diphtheria toxin or cholera toxin, which are expressed from prophages. And so this is another reason why temperate phages are generally not preferred. So that brings us into 2023. So so far, and classically, really, phage therapies rely on natural phages isolated from the environment. So this has been the standard mode, and the historical practice here has really just been you have your clinical isolates, and you go out into nature, and you get phages for those isolates, and if you can't find a phage, you go out again, and you just keep doing that. But as we've been moving along in this space, we realize now that the natural resource really is not equally abundant for all the targets that you might want to go after. So if you're going after something like Klebsiella pneumoniae or Pseudomonas aeruginosa or E. coli, there's lots of phages out there. You can go out and get virulent phages that are pretty suitable for as a therapeutic pretty abundantly in nature. But as we're finding, we go to other kind of stranger bacteria, things like Burkholderia. In our experience, Acinetobacter is like this as well. It's much more limited virus sphere. The phages are harder to find. You're looking at many samples before you get any positive hits. And all the phages that we are finding are generally not very suitable for this. I see Martha's in the audience, so she'll attest to this with Clostridium difficile is like probably the king of this problem. But there's other organisms as well. So really the idea that you can just get tons of virulent, T4-like phages out of sewage for any pathogen is really not the case. So this has led us to a route where if we want to be able to use phages for some of these infections, we're probably going to have to start trying to engineer these phages or modify them in some way to make them suitable for therapy. So this brings us to talking about Burkholderia cepacia complex. So this is a strange bacteria. It's actually a soil bacteria and was first recognized as a plant pathogen. It's also associated with very aggressive pulmonary infections in people with cystic fibrosis. And cystic fibrosis, and there's a few other immune compromising disorders where the people will become susceptible to Burkholderia infections. It generally doesn't infect healthy adults. The same strains actually can cause disease in plants and also to cause disease in humans. So this was originally described as Burkholderia cepacia. It's now recognized as a complex with 24 different species in this complex. Excuse me. Burkholderia infections are generally very difficult to treat. and they're associated with poor outcomes in transplant complications in people with cystic fibrosis. So this can be carried for a long time, months to years, before it becomes a really serious problem for the patient. But then it can, at a kind of an unpredictable time, develop into what's called Phages syndrome, which is this kind of really invasive necrotizing pneumonia, which causes massive damage to the lung and can lead to really rapid decline in function and death. It's also a real issue in transplant patients, where if they're colonized with Burkholderia, they can often carry this infection over into the transplanted lung, where it will then cause a new infection in the new lung. And this is why actually a lot of centers will actually not transplant into patients who have Burkholderia infections. So our experience has really been that phages infecting Burkholderia are relatively limited. We've received about 50 clinical isolates from CF patients in the US and Canada over the last several years. We've found that phages infecting these isolates are not very abundant. We're typically getting them from soil. Most of the Burk phages that we've identified also appear to be temperate. And the host ranges are also quite narrow. They'll infect the strain we isolated them on, maybe one other strain, and that's about it. Partly that's the phage, and partly that the bacteria we're actually trying to go after here are very diverse. So a lot of these infections are acquired from the environment, and so the diversity of the pathogen is very high. So this really points to a need for engineering these phages against the pathogen here. So at the bottom here, this is just a summary of some of the phages we've isolated so far that we've actually sequenced. So this is against Sinus hepatia, gladioli, and vietnamensis. These are 3 species in that complex. And so you can see a bunch of them are P2-like with the temperate phage. These BSEP22-like phages we worked on some years ago, and these are also kind of cryptically temperate phages, although they don't form stable lysogens. These guys here appear virulent, but they're not related to anything else, and so we don't really know much about them. And even one of the KMV-like phages we have here is actually it's a temperate version of KMV. And we published recently on temperate versions of T7-like phages, and that also appears to be a thing that can happen in KMV-like phages as well. So this brings us to a phage called Milagro. So this is a P2-like temperate phage. I was isolated against a clinical isolate called MS1. And this was collected from a post-transplant CF patient. So it's related to P2. It's more closely related to phage KL3, which is another well-known Burkholderia temperate phage, or sorry, that was actually a virulent phage. But this phage is a wild-type temperate phage, and it's able to form a stable lysogen in its host. So here's a map of it here. It's a pretty typical, P2-like phage. You have the gene modules here, the lysogeny control module here, which will be relevant, and the tail fiber and tail fiber assembly chaperone genes are right here. So the initial interesting phenotype of phage Milagro is that it forms autoplaques. And this is something you see in the literature occasionally, but hasn't really had a lot of explanations for what the deal is with autoplaquing. So this is a picture here of a lawn of the MS1 Milagro lysogen. So this had no phage added to this plate, to this lawn. So this is just the lysogen. You just plate it out by itself, and it just spontaneously generates tons and tons of plaques on the plate, anywhere from a few hundred to a few thousand plaques. So you can see if we spot the parental phage Milagro on here, you get no response. So this lawn is a lysogen of Milagro, so it's not sensitive to Milagro. But if we pick any one of these plaques off and grow them up and then spot them onto this lawn, you can see there's a clearing here. So the lawn here is sensitive to these autoplaqued phages which come off. And these are what are called true Vir mutants, as opposed to, for example, a C1 deletion. So a true Vir mutant is not able to form a lysogen, but also it's unable to respond to the repressor that's present in the cell already. So it's not repressed by its own lysogens, which is different from a C1 deletion, which is the more direct way to get a virulent phage, because those will not be able to form lysogens, but if they infect a lysogen, they'll still be repressed. So these are what we call true Vir. And this was a really good entry point then for us to be able to engineer this phage, because we didn't really have to engineer anything at all. The phage just did this for us all by itself, which was very convenient for us. So we looked at the sequence of these Vir mutants. So we sequenced a dozen whole phages and about a dozen more out of just this lysogeny control region here. So the predicted lysogenic switch here is a pair of divergent transcripts controlled by 2 promoters here called PR and PL. So this looks a lot like the P2 lysogenic control switch. You have a repressor here which is driven by this PL promoter, and you have these other genes, which we're not sure what they do, but they're probably involved in lytic function, and these are driven by PR, and they just have their own little terminator here. So this is, we're pretty sure, is the repressor. And we sequenced all of these Vir mutants that we isolated. The only mutations we ever found were in this region here, in this PR region, and really kind of between the -10, -35, and then upstream of the -35 region, where we see these individual SNPs. And so these are the only mutations we see in these Vir mutants, which then leads us to a model of regulatory activity where this is the repressor, and then that makes this likely the operator binding site. This is how this also works in P2. The repressor binds to this operator site. And so the difference here for Phage Milagro appears to be that really just a single SNP or a couple of SNPs is enough to prevent the repressor from binding to its operator. So it's very easily converted into a virulent phage. As in other phages, you need multiple mutations in order to get this kind of true Vir. But here we're seeing really just really individual small changes. So to confirm this, and also to look at what the rest of the phage was doing, we did some RNA-seq on just the dormant lysogen of Milagro. So we were looking really for this signal right here, because if this was the repressor, if gene 30 here is the repressor as we were expecting, then we would expect to see this expressed from the lysogen, and which is what we see here. The gene 30 and this other transcript, other gene which is on this transcript, which is gene 29, which we're not sure what this does, are both expressed in the dormant lysogenic state, which is then typical of a phage repressor, right? So these are on in the lysogen. And we also saw these genes down here being expressed pretty strongly, actually, from the lysogenic state. And these are our predicted morons. So morons use more DNA. That's why we talked about having these potential toxin or virulence determinants. So these, I'm not going to talk about these too much, but these look like some kind of TA system and a pretty good looking type 2 restriction and modification system. So those are the morons that the phage is carrying with it. You can see these are expressed in the prophage state. So we were interested in all these different Vir mutants that we isolated, if there's any difference in the virulence of all of them. So we did this kind of standard virulence assay. So the black line here on top is the bacteria by itself. So the blue line here, this is the Milagro, the parental, temperate Milagro phage added here on this blue line. You can see it gives a little bit of repression here, and this is not typical for what you see for a temperate phage. And we think this is because even though you infect it with this parental temperate phage, once it starts making lysogens, those immediately start producing Vir mutants, which then also start accumulating in the culture and start actually killing it also. But these are the Vir mutants added initially, and you can see they're all basically the same. This was kind of the lesson here, is that there's not a real big functional difference here between all these different Vir mutants that are generated, so we can pretty much just pick any one to move forward with. Another thing we're interested in was to see if the Vir mutants had any expansion in their host range, because one possibility is if Milagro-like lysogens are common in Burkholderias, then we have a Vir mutant, and where they would have been repressed before, now they're able to infect. So here we have a panel of different BCC isolates. The parental Milagro phage, so it infects its original host, MS1. It's able to infect a few other strains. And the Vir mutant's not a huge difference here. It's able to infect a couple of other strains, not very well, but otherwise we don't see an enormous expansion in the host range here in the Vir mutant. So this brings us to another aspect of this talk, which is the tailocin. So these are high molecular weight bacteriocins. You may also know these as what are called piacins, the R2-like piacin of Pseudomonas aeruginosa. And these are just basically a phage tail without a head. And these are antibacterial protein complexes. And the bacteria really use these for interstrain or interspecies competition to eliminate their competitors. And so these work really just as a naked phage tail. They have to absorb to the surface via tail fibers that they have. And then the tail just contracts and then basically just puts a hole through the cell wall, and the cell then depolarizes. And that's how it kills the cell. So you just have this unregulated efflux of cellular contents, a collapse of the PMF, and cell death. So these just kill by puncturing the cell wall, but these things, they don't replicate and they don't contain any DNA. So a tailocin called B-set Milo, this was isolated by our collaborator, Dr. Carlos Gonzalez and Huichan Yao some years ago. So this is originally isolated from a Burkholderia unicepacia isolate, and it was found to have a very broad host range among Burkholderia clinical isolates. once we compared its sequence to Milagro, we see it actually has a lot of similarity to Milagro. So this kind of answers one question for me, at least, where do the tail listens come from? And they're pretty obviously just derived from phages, because this is basically a headless version of a Milagro-like phage, because this is the entire tail listen locus here on the bottom, and this is the phage, and pretty much you just have the phage and you just get rid of the head and these other accessory components, and you end up with the tail listen. So the question here was, could we take the broad host range tail fibers from the Milo and put them onto Milagro to expand its host range? So you can see there's a lot of similarity right in here. This is what the two tail fibers look like. Well, this is what AlphaFold thinks they look like. This is just the 300 C-terminal residues, and this is an alignment here on the left of the two proteins. You can see they're really similar to each other. So we just picked a region here where there was a lot of sequence identity. This was our fusion junction between, so upstream here will be the original Milagro tail fiber, and downstream here will be the Milo tail fiber. So the predicted receptor binding domains of these will be these little red boxes here, which are variable regions in this alignment, and that does correspond to the region that AlphaFold predicts would be the tail fiber tip in this structure. So we constructed this chimeric phage, which has in the Milagro Vir background. So this is what it ends up looking like. We have the tail fiber, which is the N-terminal part of the tail fiber is the Milagro fiber, and then the C-terminal part of the fiber and the tail fiber assembly chaperone are from Milo. And we've replaced this and then put this into the Milagro phage, and the rest of it's unchanged. And now we look at the host range of this new phage, we can see it has expanded quite a bit. So this table has a few extra things on it now. So we have the original Milagro wild type host range and the Vir mutant. This is the host range of the tail listen B-set Milo. So the tail listen doesn't make plaques, and so the you can only really score it by kind of plus and minus. So you can see here, it's all these strains here were picked because they are considered to be sensitive to the Milo tail listen. And then here on the right column, we have the Milagro Vir Milo tail fiber fusion here. So these are all efficiencies of plating. I'm not sure if I mentioned that before. So one here, so this was then engineered and grown up in this new host here called AU41545. And this becomes the efficiency of plating of 1 for this column here. And you can see it's able to now infect a bunch of other Phages. It's able to infect some other strains down here, although the EOPs are not great. So it's able to apparently absorb to these new hosts, although there are other barriers potentially to infection here, because while they are able to infect, the efficiency of the plating aren't great. So there's still other barriers here. But you can see the host range has expanded to within the Phages. And the gist of it here really is that the addition of the Milo tail fiber to this phage has really conferred the Milo host range to the Milagro phage. So now its host range really kind of mirrors that of the Milo tail listen. And even these ones here, which are minus here, which there's no plaque formation at all, we have done experiments to show that the phage does absorb to these strains, even though it's unable to actually productively form plaques. So another thing we wanted to do to continue engineering here was to delete the lysogenic repressor of this phage. So this was done actually in a CRISPR-Cas9 system, which was developed in Burkholderia. And so we just made this phage. So this is in the Milo fusion Vir phage background, and it's really just a deletion of C1. So we did this because these lysogenic repressors, they sometimes bind to multiple points in the phage genome. In Lambda, they bind to 2 points. C1 binds to 2 points in the genome. And so if it has a second site of interaction, that could also be suppressing its activity. So we thought it would be best then to just delete C1 and see what happens. So in this case, we didn't really see much effect here. So this is another virulence assay here. So this is the bacteria by itself. The orange here is the Milo, and the gray here is the C1 deletion. And you can see maybe a little increase in virulence, but really not much. So there's not an enormous change in the phenotype here by deletion of the C1 gene. So this didn't really seem to improve virulence a whole lot, but we think this is also a good thing to do just for engineering the phage. You want to try to avoid reversion as much as you can, and really these things were only one SNP away from their original temperate state. So we want to start, deleting other lysogenic accessory functions here. So excuse me. So to wrap up, this brings us really to the future directions here. So we think that this Milagro phage is really a good platform for engineering more phages that are active against multiple BCC isolates, for example, by identification of additional tail fiber donors. And as we saw in the table before, looking at the host range, there are other potential anti-phage systems that are in these recalcitrant hosts, right, that have these low efficiencies of plating. So we still do need to sort out why the phage, for example, here it's able to absorb to these now, which it really couldn't before, but the infection efficiency is still pretty bad. So there's still other issues going on in these hosts, and it's not clear right now whether this is anti-phage defense systems or restriction or CRISPR. CRISPR is not very common in Burkholderia, so it's unlikely to be that, but probably some other anti-phage defense system or restriction system. Oops. So that's kind of the next step for us. And we're also then proceeding with deleting other accessory functions here to try to make this, a better virulent phage and lower the risk. So we're proceeding now with deletion of the integrase and of this moron region. So we actually have deleted a patch of genes here, the morons and some accessory genes, and the phage still grows fine. So it doesn't appear to be essential for phage growth. So we will be able to have a phage that doesn't have these genes in it, and that's still functional. These don't appear to be essential. So with that, I'd like to wrap up. I'd like to thank, our collaborators in the Gonzalez lab who identified the myelotalicin originally and has been doing a lot of the work here. Technician Gui Chun, our postdoc Abby Korn, and our new graduate student, former technician Hannah Peterson. And then over in the Center for Phage Technology lab, Dr. Mei Liu, Dr. Justin Beckman, Tram Lee, and Curtis Ross, who have supported the project there, and the people who have given us money to do this work. So this project is mostly supported by the Cystic Fibrosis Foundation in the US. So with that, I can close and take any questions.